Hello and welcome to our Chapter 22 video lecture series. In this video, we're covering Chapter 8 on immunoglobulins. And essentially, immunoglobulin is a fancy word for antibodies. They are proteins produced against a particular antigen. And so antibodies can tag a pathogen for destruction by immune cells. I always say it's kind of marking it with a red X for our immune cells to destroy. And so we can create a good defense against viruses, bacteria, toxins, and yeast spores. Our soluble antigens are combated by humoral immunity, which is what our antibodies work within. Now let's look at the structure first. It has this Y shape to it. Like we said, they are going to be soluble, and they're composed of 4 polypeptide chains. We have 2 identical heavy chains, which you can see right along in here. They're the same on both sides, and then 2 identical light chains, which we see over here. They're going to be flexible at this hinge region of our 2 heavy chains and held together with a disulfide bond to form an antibody monomer. Mono means 1, so this is just 1 antibody here. There are 2 important functional areas. We have the variable region, which is this area in here. You could see it contains this antigen binding site that's going to change depending on which antigen it's for. And then we have the constant region, which is all of this area in here. So as the name suggests, it will be constant, the same. So that's kind of what this slide is saying as far as the text. Let's just review really quick. We've got the variable region over here. This is located at what we call the arms of the antibody. And like I said, the antigen binding site can change. We'll bind these antigens through weak intermolecular forces. They could be through hydrogen bonds, ionic bonds, and hydrophobic interactions. Our constant region is going to contain the FC region, which is the stem, so this portion right in here. And this area really determines the biological function. It's going to be the same in structure for the antibodies of a given class. So in a little bit, we'll kind of talk through the 5 major classes. We've got IgG, IgM, IgA, IgD, and IgE. And each one kind of has a different specialization. But before we get into that, let's talk about what the actions of our antibodies are. Essentially, it has 6 different ways in which it can cause destruction for the antigen. So first is going to be neutralization, and this is when we physically cover the antigenic determinants, which are the teal spheres here that we see, and we are making it ineffective in establishing an infection so that it can't really bind to anything else or cause destruction. Then we have agglutination, in which the antibodies are going to cross-link. Notice how this antibody is attached to an antigen here as well as over here. So the cells are now going to be clumped together, which doesn't allow it to cause damage. Then we have precipitation, and this is when our antibodies are going to cross-link circulating antigens, for instance, maybe some viral particles, and forms an antigen-antibody complex that becomes insoluble and precipitates out of the body fluids. Precipitated complexes are engulfed and then eliminated by our phagocytes. Then we have our complement system. Specifically, our IgG and IgM are going to bind and allow for complement activation. So this is mostly within our classical complement activation system. Then we have opsonization. This is when we are going to have the Fc region of a certain antibody class make it more likely to be seen by our phagocytic cells. You could see that it kind of attaches right in here, whereas the variable region is attached to the antigen itself. Then our phagocytes, of course, are going to engulf this complex and gobble it up. Then we have activation of our NK cells. This is when we have the Fc region of some of our antibodies, like IgG, trigger the NK cells to release its cytotoxins, those perforins and gramzymes, and that is going to cause a destruction of any abnormal cells through what we call ADCC, antigen-dependent cell-mediated cytotoxicity. All right, now let's get into our 5 classes of immunoglobulins. First up, we have IgG. This makes up majority of our antibodies in our blood, so it is usually referred to as our serum antibody. It is also found in other body fluids such as the lymph and cerebrospinal fluid, and can participate in all types of antibody actions, the ones that we just talked about. What's unique about IgG is that it can cross the placenta, which is a good thing because it helps to give the fetus and infant some immune abilities, but it can also cause hemolytic disease of newborn. Then we have IgM, which is found mostly in blood as well. It normally has a pentamer structure, and it's most effective at agglutination and binding complement proteins. It's responsible for the rejection that we have in mismatch blood transfusions. Then we have IgA. This is found in areas exposed to our environment. We usually call this our secretory antibody because we find it within a lot of the secretions, like in our mucus, saliva, tears, breast milk, and it really helps to protect our respiratory and GI tract since it is found in mucus. It is going to be either a monomer or it could be found as a dimer that is composed of the 2 antibody molecules coming together. It helps prevent the pathogen from adhering to and penetrating epithelium, and it's really good at agglutination. Then we have 4th is IgD. This, we aren't too sure exactly what it does, but we know it functions as an antigen-specific B lymphocyte receptor, so a BCR, and it identifies when immature B cells are ready for activation. Lastly, we have IgE, and this is usually formed in response to parasitic infections or allergic reactions. Otherwise, we have a low rate of synthesis of this immunoglobulin. It can cause a release of products from basophils in our mast cells, namely histamine being released, and it can attract eosinophils, especially when we have that parasite infection taking place. Now, we can have class switching take place, which is when our plasma cell is going to change the type of antibody that it produces. This does require contact between our helper T cell and the plasma cell, and our T cell has to release certain cytokines to specify which antibody class has to be formed.